Now it is working. So let me just, uh, can you see my screen?
Yes, sir.
Okay. Okay. So good morning. I'm going to start a new chapter. It is about the metabolism. And so far you have studied about the individual macromolecule. You have learned about the specific functions of the molecule. You have looked at how the cell can transport the molecule across its membrane. Now we are going to move on to a network of reactions which are happening inside the cell. So we are going to talk about metabolism. Here we are going to focus on basic concept and design. And so the first question comes for any biochemist is that how does a cell extract energy and reducing power from its environment? That means if a cell is growing in an environment, how it gets its own energy from the environment. And number two question is that how does a cell synthesize the building block? For example, I have to synthesize protein. That means how a cell is going to synthesize amino acid first, and then from these amino acid how it makes its macromolecules. Then these are generally carried out by high network of reactions, and these are chemical reactions as you have seen in individual reactions. These reactions are chemical reactions that are collectively known as a metabolism. So if I want to typically show this metabolic reaction, it's a very complex reaction. You can see that there are several reactions which are going on to give you a network of reaction. I'll come back to this again. So let us continue with the discussion. So the metabolic processes are the one which answers the question about how a cell extracts energy and how these energy, when it is being extracted, is being utilized for the synthesis of building block. So if you look at this, there is a coherent design in the system. That means the cell is carrying out these processes by a certain design, and that is where we are going to talk about metabolism, basic concept, and design. Approximately around 100 molecules, they play a central role in all the forms of life. You take from microorganism to higher organism. These are some 100 molecules, they are playing important role. We have talked about four macromolecule, but there are other molecules. You see that if I compare with organic chemistry and the biological chemistry, you find that there are thousands of organic molecules and they are carrying out different reactions. Here, what you see is that the number of molecules are relatively less, but they carry out a lot of reactions. So that is what we are going to talk about. The number of reactions in the metabolism is large, and the number of a kind of reaction is small. So on the other day, I have talked about enzymes. We have talked about six different types of reactions. So you can broadly see those six types of reactions which are happening in the biological system. So the process and any metabolic activity starts from the energy, because unless you have energy, you cannot carry out the reaction, you cannot perform the reaction, or uh, these activities, and that's why it is important that cell transforms different forms of energy. So here the living organism, they require a continuous input of energy, free energy, and there are three main purpose of this energy. First one is to perform the mechanical work in muscle contraction and other cellular movement. So whenever bacteria is moving, whenever our muscle is moving, they require energy. So we'll talk about from where this energy comes. Second thing you have seen is that whenever the membrane transport happens, whenever the molecules they are moving across the cell and it is against the concentration gradient, you require active transport. And it is not only molecule like carbohydrate, like proteins, like smaller sugars and amino acids. It is also for small ions. So even if ion has to move across the membrane and it is against the gradient, you require energy. And the third thing, which is the most important thing, is that they synthesize macromolecule and other biomolecule for simple precursors. So that means you require energy when you synthesize protein starting from amino acid. You require energy when you synthesize the carbohydrate, which is a polysaccharide from monosaccharide. So that is where the energy is being required. Now, question is from where this energy comes. The energy comes from two different major sources. One is the solar energy, which is a master energy, which is the major energy source. And this light energy is being utilized by photosynthetic organism, and we call them as a phototroph. So whether it is a phototrophic organism belonging to the bacteria or belonging to the plants, they are the one which are transforming the light energy into chemical energy. So you look at the transformation of energy. The solar energy or the light energy is being converted into chemical energy. So what is this chemical energy? Can you guess? In plants, have is that they are synthesizing simple carbohydrates. So the fixed carbon dioxide. So if I take carbon dioxide and water, what you can do is that you can make carbohydrate. So that's the way the energy, which is a light energy, can be conserved in the form of chemical energy. And this chemical energy, when is being utilized by chemotroph are the one which are utilizing or which are obtaining the energy from chemical compounds. So here, the chemotrophs, what they do is that they use energy by oxidation of foodstuffs generated by phototroph. So phototroph, like plants, they synthesize carbohydrate, and those carbohydrate is consumed by chemotroph, and that's the way it is being utilized. So what it means is that whenever your carbohydrate is synthesized, whenever the bonds are being formed, you require energy, and this energy is coming from the light energy. And once these bonds are being synthesized, in the next step by chemotroph, they are being utilized in conversion of either another type of chemical bond, that is what you have in ATP, that is the source of energy, or they are broken for carrying out other reactions. So it's a basically we are looking at the network of reaction which we are talking about. So the chemical energy obtained by the oxidation of a carbon compound, here major carbon compound could be carbohydrate, may be transformed into the unequal distribution of ions across the membrane. So what it means is that we derive the energy, then this energy can be used for transport of ions. It could be transport of other molecules as well. Now, that means that if I transport ions across the membrane using that energy, which you have seen in the membrane transport classes, and this energy creates a gradient. And if gradient is created, what will happen? This will carry out other transport. So the primary transport and the secondary transport. So ion gradient are the one which drives the secondary transport. You have learned this, like sodium ion going in, helping the other molecule to enter in the cell, or one ion entering inside the cell, allowing the another smaller molecule to enter inside the cell. So these are the process. So the gradient once created by this chemical energy is going to help you in transport of other form of energy. So now question comes that this chemical energy can be employed in different form. It could be even for transfer or convey information in the form of nerve impulse. The chemical energy can also be transduced into mechanical energy. This is very important. For example, bacteria, if it moves, it requires energy. If our muscle is moving, it requires energy. So what it does is that this chemical energy, which is in the form of some carbohydrate or any other protein or anything, when the bonds are broken, the energy is transformed from chemical energy to mechanical energy, and this energy is doing structural alteration in proteins. So there are contractile proteins, that actin and myosin present in the muscle. They will result in muscle contraction and the movement. Now, we have to sum up this slide, which says that energy is being extracted from the fuel, and used to power the biosynthetic processes. So the mean is that the question which I have asked in the beginning, that from where we are extracting energy. So we are extracting energy from solar or light energy, and then it is converted into chemical energy by phototrophs, and these chemical energy is being utilized by chemotrophs, and then the chemotroph uses this chemical energy for various other transformations. And all this network of transfer of energy, the reactions which are forming energy or the reaction which are utilizing this energy are termed as a metabolism or intermediary metabolism. Now, coming to the interesting concept that how this metabolism occurs in general. So the metabolic pathway we can divide into two broader classes. And as you have seen that in some reaction you require energy, in some reaction you produce energy. So those reactions which convert energy into biologically useful form, so that's a one type of metabolic pathway, and another one where you require input of energy to proceed. So for example, if muscle action occurs, you require energy. For example, if you want to transport a molecule, you require energy. So all these require energy. But on the other hand, if I want to produce energy, you can break down carbohydrate and you can have energy. You can break down proteins, you can have amino acid that can enter in a pathway, you have energy. So there are different ways in which energy is being produced, and there are different ways in they can be utilized. So those reactions that transform fuel source into energy are called as a catabolic reactions or catabolism. So catabolic process, kata means breakdown. So catabolic process are the one which breaks down the complex molecule, could be carbohydrate, could be fats. Mostly it is carbohydrate and fat, but even protein degradation can produce energy. So once they are broken down, they are converted into similar molecules. So you have C6H12O6 that can produce CO2 and H2O. And what you have generated is you have generated useful energy. So they be trapped in a specific form that we will discuss, which is ATP. But what you learn here is that there is a process where a complex molecule is broken down to simpler molecule, and during this process, the chemical energy is being released, and that is what is preserved by the cell. Now, second type of reaction is the one which is utilizing useful energy into a small and this energy which was produced in the first reaction is going to be useful with the small molecule, like we have amino acids. Now what will happen, the energy which has been produced in the first reaction will be utilized, and this energy can synthesize a complex molecule, that means polymers. So am I audible? Hello?
Yes,
Can
sir.
you hear me? Yeah, and the slide is visible. Okay, let us move on. So here, what you are looking at is that catabolic reaction is converting the complex molecule into simpler molecule plus energy. On the other hand, the energy produced in the first form, that means catabolism, is going to be utilized for anabolism. Ana means synthesis. So you are looking at the synthetic processes. So you have a set of reaction which is called as the catabolic reactions or catabolism. Then you have another set of reactions which are called as anabolic processes or anabolism. So you are utilizing the useful form of energy that are produced in catabolism for the anabolic reaction. But in some pathway, you will find that both the reaction occurs. There is a control. Whenever you have excess of energy, what it will do is that it will go for anabolic process. But on this other hand, when you have less energy, this will go, the same reaction will go for the catabolic reaction. So you have both catabolic and anabolic operation in the same set of metabolic pathway, which I will talk when we will talk about the Krebs cycle or TCA cycle. Now, just to give you a glimpse of the type of reactions which are occurring, you have a central theme where energy is being generated, that you have the TCA cycle, which produces energy, and this energy can be utilized for different reactions, for synthetic reactions. Then you have several complex carbohydrate and other molecules which are catabolized, and when they catabolize, they make a central theme of a molecule which is going to produce all forms of energy. So slowly you will learn that whether it is a carbohydrate, whether it is a fat, or whether it is a protein, all of them metabolize and make a central molecule which enters in a simple pathway, same pathway, and most of energy is produced from that reaction. This is an interesting concept which is utilized from smaller microorganism to higher microorganism. Now question comes, how this reaction occurs? Do we have all the reaction which are thermodynamically favorable? The answer is that even if in a metabolic process, if there is a reaction which is unfavorable, they can be driven by a favorable reaction. So let us talk about this concept. I will give you some simple example and explain it to you. So a pathway, and the question also comes that how do we define a pathway? So any pathway in metabolic reaction can be given by the individual reaction, which should be specific. For example, if I talking about the glycolysis or EMP pathway, what we are talking about is glucose should be converted into glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, to fructose 1,6-bisphosphate, to your, and so then they are being broken down. Fructose 1,6-bisphosphate is broken down. You get two molecules, they again enter in the pathway. So you have a set of reaction which are specific reaction. And the entire set of reaction that constitute the pathway must be thermodynamically favorable. So suppose glucose is converted into pyruvate, you will find that though in between some of the reaction may not be favorable, but at the end, conversion of glucose to pyruvate should be favorable. What it means is that the delta G of the sum of the reactions of all the reactions present in a pathway should be negative. So delta G should be negative. Now, coming to a thermodynamic reaction, as you know, free energy is a very, very important concept. And it holds true whether you are talking about chemical reaction or you are talking about the biological reaction, which is happening inside the cell. So what happens, this delta G of a reaction depends on the nature of a reactant and product. What it means is that in this reaction, if I'm going to calculate delta G, it is not dependent on standard free energy change of free energy. It is not just standard free energy change. It also involves the concentration of the product as well as a concentration of your substrates. So it's a very important concept. So if any reaction which is happening in the test tube, and if it is happening inside the cell, if the concentration of your C and D, that means the product, and concentration of reactant is different in two different environments, you will find the delta G will be different. This is a simple concept, nothing great, but what is important here is that one must know that how much concentration of the product and substrate are present in the cellular environment. That helps in calculation of delta G. So this delta G value may be, different from the standard free energy change, which is known under given set of conditions. So an important thermodynamic fact is that overall free energy change for a chemical coupled series of reaction is equal to sum of the free energy change of individual step. This concept I'm going to discuss in the next slide. Here, what it means is that suppose you have first reaction, which is A is converting B plus C, and second reaction is a B converting into D. First reaction is having delta G positive. Now, this reaction should not be feasible. That means A should not convert B into C. But somehow, if B is converted into D and delta G is negative, and the sum of these two reactions gives us delta G negative, that means A will be converted into C and D. So this becomes the thermodynamically feasible reaction. Now, thus, a thermodynamically unfavorable reaction can be driven by thermodynamically favorable reaction. It's very important that because This type of reaction occurs inside the cell as well. That means they derive a reaction which is not thermodynamically feasible. So your metabolic pathways are formed by coupling of enzyme catalyzed reaction such that overall free energy of the pathway becomes negative. Now, from where this energy comes, what can be couplers? So that is what we are going to talk about. So ATP is the one which is having high energy, and from where this high energy comes, that part I'll discuss separately. But it is important that you should know that ATP is the universal currency of free energy. We will also talk about why ATP has been chosen as a universal currency and what is unique in ATP, that is why it derives other reactions. So it is basically whenever we try to transact, what you have rupees. You give rupees, you can buy something. So here ATP is the one which becomes a common energy currency. So whenever you have excess of energy, ATP will be synthesized, and this ATP is the one which will have energy, and this energy can be given freely to another reaction wherever it is required. Now, looking at the chemical form of the ATP, ATP is the one which you have already studied under the nucleotides. You have adenine. You have learned about the nucleotide, and in nucleotide, what you have learned about is AMP, adenosine monophosphate. Or so you have the base, which is adenine. You have a sugar molecule, and the sugar is a ribose sugar, and you have a phosphate. Now here the phosphate groups are not single one. There are three phosphate groups which are associated. So I will talk about the importance of those three phosphate groups. And in most of the reaction, you will learn that the active form of ATP is usually complex with the magnesium and manganese. So these are the ones which are helping in the complexion or binding and those events, as well as there are a lot of reaction where the enzyme, they will require these ions. The ATP is the energy rich molecule. And why it is so? Because triphosphate unit contains two phosphoanhydride bonds. I will further explain that what actually energy means. Does it mean okay, so I'll pose a question and I would like to learn from you. If I say ATP is a high energy rich molecule, what does it mean? Any one of you? Normally, general wisdom is that if I have a molecule and if I have to break a bond, I should spend energy, isn't it? Okay, let me see if something. Can you hear?
Yes, sir.
Yes, sir.
Okay, okay, fine. So, did you get the question which I am talking about?
Yes, sir.
Yes, sir.
Okay, so normally what happens, suppose I have a and I break them. I have to break a bond. Conventionally, I should think that if I have to break something, I will require energy, isn't it? But on the other hand, I'm saying that when ATP is hydrolyzed, you are getting energy. So what does it mean? What actually it means? Okay, think about it. Within a few minutes, we are going to discuss about it. So what I'm talking about is a large amount of free energy is liberated when ATP is hydrolyzed to adenosine phosphate. So ATP converted into ADP, you get a lot of energy. ATP can also be converted into AMP, adenosine monophosphate, where you get pyrophosphate, and pyrophosphate can also be further, broken down. So you can have more energy when the PPI bond is broken. So looking at this, uh, can you see these molecules? You have ATP, the structure of ATP, ADP, and AMP. On the terminal residue, you have three phosphor group which are present, and they all have a negative charge. So what we are going to talk about is some of the biosynthetic reactions are driven by the hydrolysis of this ATP, In some of the cases, when we talk about the reaction, you will see that GTP or UTP or CTP can also play a role. So it is not the domain of ATP alone, it could be any one of the nucleotide triphosphate. But most common one is ATP. In few reaction, you will learn that GTP is involved, in some reaction UTP is involved or CTP is involved, but they are rare reaction. ATP is the most common currency. And the diphosphate will be, of course, once you remove one of the terminal phosphate residue, you will get GDP, UDP, and CDP. And if I remove both of them, we'll get GMP, UMP, and CMP. Now, most of the cases, whenever we are talking about the energy is being calculated, whether one phosphate bond has been broken or two of them has been broken. So let us talk about why this is important. So look at the compound. It is similar thing which was initially present in your nucleotides. Or this is basically a nucleotide. Why? Because you have a base. This is a base, which is adenine. This is the ribose sugar, and 1, 2, 3 phosphate residues. So what you see here is that once it is converted into here, into ADP, the terminal bond is broken, and then energy is being produced. Actually, what happens here, the last bond between the phosphate to phosphate residue is the one which we called as a potential of transferring phosphate group. So actually, when we talk about the energy production, it is the ATP's ability to transfer its phosphoryl group to another molecule. So this is called as a phosphoryl group transfer potential. So in an actual form, we talk about that the phosphoryl group transfer potential of ATP is quite high. So let us see how high it is. So ATP has a higher phosphoryl group transfer potential. That means phosphoryl group can be transferred compared to glycerol 3 phosphate. So you look at first reaction, ATP hydrolyzes, delta G0 prime is minus 7.3 kilocalorie per mole. And if I take a glycerol 3 phosphate, where your phosphate is again present, phosphoryl group is dissociated, and delta G prime value is minus 2.2 kilocalorie per mole. So why this difference in these reactions are occurring? So delta G0 depends on the difference in the free energy of the product and the reactant, and the structure of both ATP and hydrolysis product ADP and PI. So what is determining it? One is the structure of ATP and another is structure of the product. So product of substrate and product of reactant. Now the factors which are responsible are, one is the resonance stabilization, which molecule should be producing energy or stable, which can have which molecule, whichever the molecule can stabilize by resonance. So if you look at the reaction, the PI which is being produced in the reaction can be resonance stabilized because it can have different forms. So if you have a phosphate group, you have OH on one side, H on the other side, double bond O is on one side. Now this can be with different forms. So now this PI can exist in different forms. I'll show it to you. Then to product ADP and PI. So depending on the structure, that is important. Second thing is electrostatic repulsion. If I look at the ATP molecule, as you have seen in the previous slide, this repulsion is much higher than this repulsion, or here you don't have the repulsion. So which one should be more energetically tending to transfer its group? So ATP will be the one which will be trying to transfer its terminal phosphoryl group to the ADP, to another molecule to become ADP. Then third one is the stabilization due to hydration. So all these points, all these 3 points, that is resonance stabilization, electrostatic repulsion, and stabilization due to hydrations, are concerned when we talk about their ability to transfer phosphoryl group. So if you look at the ATP and ADP, you find that ADP and particularly PI have a greater resonance stabilization than reaction will move in the forward direction. Second thing is that the orthophosphate group has high energy whereas the gamma phosphoryl group of ATP has a smaller number of there. So if I look at the number of forms, if I look at orthophosphate, I can make different resonance stabilized forms. Whereas if I take ATP, the number of forms are limited. This is the one which shows you resonance stabilization. Phosphate orthophosphate group can exist in all the four forms, whereas the resonance stabilization of a terminal phosphate group is limited. So at pH 7, the triphosphate unit of ATP carries about 4 negative charges. These charges, they repel each other, and because of their close proximity. So it is a electrostatic repulsion which is happening. The repulsion between them is reduced when ATP is hydrolyzed. So the reaction will tend to form or will tend to hydrolyze ATP into ADP and PI, and that's why your delta G is negative. That's number one. Number two is the water can bind more effectively to ADP and PI than it can to the phosphoanhydride part of ATP. So if I look at the phosphoanhydride part of ATP and I look at the ADP and PI, you can see the water can bind more effectively to ADP and PI, and that's why the reaction will tend towards the formation of ADP and PI, and that's why the delta G is negative. Third reason is that ATP is often okay. So you have seen the resonance stabilization. You have seen that ATP is not, not stable form in terminal group. You have seen that there is electrostatic repulsion, and you have seen hydration. In all the three forms, you have learned that ATP tends to be converted to ADP and PI, and that is where you have delta G negative. Now, in general, this high phosphate group or phosphoanhydride bond are referred as a high energy bond, and in representation it is given by squiggle, which is denoted by this P. And whenever this P is denoted with a squiggle, that means that particular bond has high energy. That means they will have a tendency to break. Now, coming to the question, that first thing is that why ATP has been chosen as a currency of energy transfer, and number two, can we transfer phosphoryl group from other molecule to ATP? In other words, can we synthesize ATP? Or the second thing is that can we, first thing is that can we synthesize ATP, and second thing is that can we, hydrolyze ATP to produce energy. So first part you are very clear. The second part is that there are certain compounds, and they are not very many. There are very few compounds which can transfer its energy or its phosphoryl group, or they have a high phosphoryl group transfer potential than ATP. So these compounds are, for example, phosphoenol pyruvate. So if you look at the biological reaction, these compounds are the one which will be involved in the ATP synthesis from substrate level. I will talk about later what is a substrate level phosphorylation. That means substrate has a higher energy so that ATP can be produced. So these three molecules are typical molecule, for example, phosphoenol pyruvate, for example, this 1,3-BPG and creatine phosphate. So these molecule can transfer its phosphoryl group to ADP to form ATP. And ATP has a phosphoryl transfer potential that is intermediate among the biologically important phosphorylated molecule. What does it mean? It means that most of the reaction which is occurring in cell, they will be requiring energy where your delta G will be having plus 4 kilocalorie per mole. So now what you require? You require that you should reverse these reaction by ATP hydrolysis. And how you can do that? You can, and why you can do is that because you should not have a currency which should be having storing very high amount of energy or very low amount of energy. So I'm discussing this point that ATP has a phosphoryl group transfer potential that is intermediate among the biologically important phosphorylated molecules. So you will see typically all the reactions which are happening they require energy in the range of 4 kilocalorie. That means most of the reaction they will have the plus value if it is reversible, if it is irreversible reaction, or if it is having delta G plus value, that is something in 4 kilocalorie per mole. It is not very high, not very low. So what ATP does is that it has to have energy which can carry out most of the reaction if the delta G is a positive. So if you look at the central theme of the metabolism, ATP can be employed for most of the reaction where they are not feasible reaction because of the thermodynamic delta G positive value. On the other hand, if I think, so you may say that why ATP is not having, why we don't have a creatine phosphate, or why don't we have some other molecule which has a very high energy which can take care of all the metabolic reactions requirement. Answer is that if I have here itself in the case of ATP, you have seen that it is minus 7.3 kilocalorie per mole it is required. It is the delta G value. So now what happens whenever such reaction happens, some energy is going to be wasted. So where does this energy go? They become as a heat energy. So whenever it is a coupling, some amount of energy is lost, but still the loss is less. Suppose I have, instead of minus 7.3 kilocalorie, if I have minus 12 kilocalorie per mole or minus 20 kilocalorie per mole, lot of energy will be wasted. So that is why ATP has been chosen by the system, so that it can take care of, it is of intermediate range, it is something which is in metabolic reaction, its delta G value is in intermediate, and it can be utilized for the other reactions. Now, some molecule like creatine phosphate, they have a much higher energy or much higher delta G value, so they can transfer its phosphoryl group to ADP so that ATP can be synthesized. All these reactions, if you look at, they are driven by chemical equilibrium, and this is why, because the concentration of ATP and ADP and creatine phosphate, they derive the reaction. This is another, slide which is showing what is the different types of molecule which are giving energy. So you have phosphoenol pyruvate, creatine phosphate, and 1,3-bis phosphate, bis glycerophosphate. So all these reactions are the one which are going to give energy when they are being when their phosphoryl group is transferred to ATP molecule. So they will be helping in the synthesis of ATP molecule. And once ATP is synthesized, then you can use them for the general metabolic reactions. So I'm stopping at this point for a few seconds, and let us see if you have any questions. Any question? Any one of you? But then what you have learned, let me just repeat. First thing you have learned that the metabolism is consisting of both catabolic process and anabolic process, and both of them combined together are called as a metabolism. We have also talked about that there are different types of transformation of energy, and this energy is transferred from one form to another form, and actually the source of all the energy is light. From the light you synthesize the carbohydrate, that means you get the bond energy. So for example, you have a carbohydrate. So say I talk about C6H12O6, which is a glucose molecule. Each bond will be having, is contributing towards the production of energy. So when it is a completely catabolized, when it is a completely converted into carbohydrate, CO2 and H2O, you get a lot of energy. When it is a partially converted into pyruvate, you get little bit energy. So you look at the energy produced is a function of the catabolic reactions. Another thing you have learned is that these chemical energy can be in different form. One is the which is we are talking about carbohydrates, where you have a bond energy. Then this bond energy is converted into production of ATP, which is a universal currency. So why we talk about the universal currency? Because it can, take energy from very high energy rich molecule like creatine phosphate and uh, the other molecules like phosphoenol pyruvate. On the other hand, they can, give the energy to the other reaction. So that's why it is a universal currency. Now, the type of energy transformation you have learned, it could be mechanical energy, chemical transport energy, etc. And you have also learned that how these interconnecting reactions are involved in the, actually utilizations. You have learned the concept of catabolic reaction. You have learned about the concept of anabolic reaction. You have learned that there is a huge chemical network which is leading to the different reaction, and they are interwoven, they are interconnected with each other. And most of these reaction, as I look at to analyze all the reaction, I will find that most of them, if it is it is, non-feasible reaction, they require energy input of 4 kilocalorie or so. And that is why the delta G of ATP hydrolysis has been chosen, which is the intermediary in set of the molecule, which are biomolecule having high energy capability. You have seen that the unfavorable reaction can be converted or driven by favorable reaction. You have also learned this is the ATP, which is universal currency. I am giving you this reaction. Why? Because when ATP is hydrolyzed to ADP and PI, you get delta G, which is minus 7.3 kilocalorie. If I look at, and this is under standard set of conditions, so I'm giving you delta Z zero prime, this is under standard set of condition. But if I look at this reaction inside the cell, where typically the concentration of ATP, ADP, and PI has been taken into consideration, they are going to be minus 12 kilocalorie per mole. So there is a slight difference which is occurring between the standard free energy and the cellular condition under which we are calculating delta G for ATP hydrolysis. When we talk about the hydrolysis of ATP to AMP and PI, you will have much larger energy. That means delta G is slightly higher than the hydrolysis of ATP. I have given you the reason why this ATP is universal currency and why it is the one which is showing the energy. Then the structural basis you have learned because of the electrostatic repulsion, the hydration, and third one is the resonance stabilization. Okay, and you have also learned that why typically some of the molecules, they are high energy rich. So for example, in muscle, if I require quick energy production, quick ATP formation, it's very difficult for the cell to metabolize carbohydrate and convert them into the pyruvate or convert them into CO2 and water. So quickly, if it requires energy, it will be synthesized from creatine phosphate to ATP. So it's a phosphorus group will be transferred to ADP and ATP will be synthesized and ATP will be immediately utilized. The next class will talk about how much ATP you actually require. The ATP is not being stored. Most of the time, ATP is synthesized during the time when you require them. So it is a very controlled mechanism which is existing in the cell where it typically controls how much ATP should be converted into ADP and PI. So energy is being utilized. And at the same time, you will see the synthetic process. So there is a huge coordination between the process where ATP is being utilized and the process in which ATP is being produced. So I'm stopping at this point. Let me know if you have a question. Any question from your side?
No, sir.
Okay. So, okay, I'll stop at this point. I'll sign off. Then we will further continue with one more class with the intermediary metabolism, how the how many what is the typical the requirement, how many of these reactions are required, what are the mechanisms. So one thing you have very clearly understood, that the ATP is the one which is universal currency, it has a high energy, and that you will be learning in the different reaction. Okay, then thank you very much. I'll sign
Sir,
off. Yeah,
uh,
yes,
sir,
please.
when will the quiz happen? Like in the course plan you mentioned there would be
Yeah, I'll
30.
not give you the date because I see that many of you are not coming on regular basis. So if you don't come, you may miss it. I'll tell you, it will be surprise quiz in one of the classes.
And sir, you also mentioned some assignments type in that only quiz slash assignments.
Yeah, so both will be given. You will be given sufficient time to do so. Both for
Okay.
assignment, as assignment you will get time. For quiz, it will be based on whatever is being taught. So maybe from previous couple of classes, you can have a quiz. Okay.
Okay, sir.
Okay, so today I'm going to have a discussion as I have already given you, I have sent the notice that we can discuss the question paper which has been given to you. So many of you may have questioned why I have not got full marks or why I have got only half marks. So I'll give you the actual answer. Many of you have written the answer, but it is not perfect answer. So that is why you have lost some marks. So we will discuss the criteria, how the evaluation was done, and what was expected from you to write. So today evening, I have given you the time slot. We will meet on the same channel in the Microsoft Teams. Okay,
Thank
then.
you, sir.
Thank you. Thank you very
Thank
much.
you, sir.